here we have a simple diagram of a muscle fiber. And here we have the sarcolemma, which is the same as the cell surface membrane. It's important to keep in mind that the muscle fiber is multinucleated. And as we have inside here, the muscle fiber, we have myofibrils, which are these. And here we see that we also have on top of them the sarcoplasmic reticulum.
This is the muscle fiber.
And then inside of these, which are here and over here, we have myofilaments. Okay, and it's important to keep in mind also that the sarcoplasm is the same as the cytoplasm in a regular cell. Here we have a lot of mitochondria inside the muscle cell because we need a lot of ATP for muscle contraction. So here we go down, and here we have our myofilaments that are made up of actin and myosin. And then here we have our myosin, which is a thicker filament, and our actin, which is a thinner, our actin, which is a thinner filament. And here on our band A, we have both actin and myosin. And here at zone H, or band H, is just myosin. We also have our Z line, which is just the actin filament. And then it's important to remember that from Z line to Z line, it's one sarcomere. Okay, so from Z line to Z line, it's one sarcomere. And here on our I band, this is where we have our actin filaments. And then it's important to keep in mind that the globular heads in myosin, when they're contracting, they contract towards the M line. That's the middle here.
Go.
So then here we have our neuron, and action potential travels down the neuron, and it causes the calcium voltage gain channels to open up and calcium ions diffuse into the neuron, and this causes the neurotransmitters inside these vesicles to fuse with the cell surface membrane of the neuron, and then they come into the synaptic cleft, which is here, and then they diffuse and bind with the active site. Okay, so it binds to the protein receptors here on the sarcolemma, which is basically the cell surface membrane of the muscle cell.
Go.
Okay, so this binding causes the sodium channels to open up, and sodium diffuses into the sarcolemma, which depolarizes it. And this impulse travels down to the T tubules. So as the tubules go down, as the impulse goes down the T tubules, it's so close to the sorry.
Yeah.
Okay, and this is where troponin comes in. Okay, so the impulse travels down the T tubules, and because the T tubules are so close to the sarcoplasmic reticulum, it stimulates the calcium channels to open up and diffuse calcium out into the cell. Okay, so what the calcium does is it binds to troponin. Here, in our little diagram here, it's over here. It binds to troponin and tropomyosin, so it changes shape and it exposes the binding site, which is this. Now, it's important to remember that calcium only binds to troponin, although it does change both their shape. And then here it exposes the binding site of actin, and this allows the globular head over here in myosin to bind to actin and cause a contraction. Okay, so the globular head of myosin acts as a ATPase. It hydrolyzes ATP, or hydrolyzes ATP, and what this does is it breaks down ATP to ATP, ADP, and inorganic phosphate. And now, as we have here, the globular head pulls back, and this is because of inorganic phosphate. And at this stage, inorganic phosphate has been released, and all we are left with is ADP. And then when ADP is released, it goes, the myosin head, the globular head, goes back to its resting position to grab onto another binding site and complete the process all over again. And now when these contract or pull back, they pull back towards the M line, and the sarcomere becomes shorter due to the contraction.